When we look at the effects of inhibitors which inhibit the clutching-mediated pathways, we saw that cadaverine and chlorpromazine did not change the flux rates of these particles across the cells. Rather than we use our positive control, which is transferrin and known to go through clutching-mediated pathways across the alveolar cells, we saw that these inhibitors decrease the flux rate of transferrin significantly with no significant decrease in the flux rate of the particles, suggesting that these particles do not use clutching-mediated for pathways either. Again, I did a colocalization to make sure to confirm the previous data, and we saw that these particles here is shown in the red, the carboxyl-modified particles do not colocalize with clutching heavy chain, which is the known marker for clutching-mediated pathways. As other inhibitors, we use actin depolymerization agents such as cytochalasin D and leutrophilin B, which are known to inhibit macropinocytosis and phagocytosis, and also this literature showing that basically actin is involved in all the endocytic pathways, not only just macropinocytosis and phagocytosis. But as I have a negative result that's showing that these inhibitors does not affect the transport of the PMP, so I confidently can go, it's not going to macropinocytosis and phagocytosis, and also most probably other endocytic pathways. As another evidence, we look at the colocalization profile of these particles with early endosome marker, E81. Most of the route of entry through the endocytosis are known to end up in early endosome. When we look at the colocalizations of these nanoparticles with early endosome marker E81, we saw that these particles here shown in green do not colocalize with E81. Consistent with all the data, I showed that these nanoparticles do not use any major known endocytic pathways for transport. Because we had this huge difference between flux rate of positively charged particles and negatively charged particles, we just thought the alveolar epithelial cell is known to have a negatively surface charge. Just wanted to investigate and see if we try to cover this, overcome this negatively charge of the surface area, how does it affect the flux rate of these particles? We use polycationic peptides such as polylysine and portamine, which are known to cover negatively surface sites of the alveolar cells. And we saw that in presence of these polycationic peptides, the flux rate of the PMP, positively charged PMP, modified, decreased between 30 to 50% and did not affect the negatively charged particles. But still the ratio of the flux of the positively versus negatively charged particles were about 15 times difference than positively charged, still going 15 times more. And also we use germ agglutinin, which is the known marker going through adsorptive endocytosis. And we saw for something that's going through adsorptive endocytosis, the presence of these polycationic peptide decreased their flux by about 90%. So we conclude based on this result and also other colocalization study that we did, that interactions of these positively charged particles with the negatively surface charged sites on the cells promote the uptake of the positively charged particles, but they're most probably not using any endocytic pathways, including adsorptive endocytosis. We wanted to come up with the more simpler models to understand how these particles interact with the plasma membrane. So we came up with these, it's relatively new, we are doing this work, came up with these models of making simple lipid bilayers on the filter, trying to mimic the components of plasma membrane, alveolar epithelial plasma membrane, and we look at the transport of these particles across this lipid bilayers on the filter. And it's interesting enough, the positively charged particles go 30 times faster across the lipid bilayers than the negatively charged particles. And what is that? That's what we don't know, and we are after right now. But what these results show is that these nanoparticles can, in the absence of any energy source, translocate through a given plasma membrane, the artificial plasma membrane. So in summary and conclusion, I would just want to show that we showed that these nanoparticles in the baseline condition when the cell monolayer is not injured cannot go through the junctions. I showed that with the results of EGTA and the cofocal macrograph that I showed. When we investigate the effects of different endocytic pathways, with the different pharmacological agent and the photon microscopy, we showed so far that these nanoparticles do not go through any major endocytic pathways. Is it possible that they dissolve through this plasma membrane and go through the cells, or they make the holes? This is the part of research that we are after right now and want to go after that. I just want to acknowledge people who helped, my advisor, Dr. Crandall, my co-advisor, Dr. Kim, lab mates, and other people who were really useful in this research. Thank you.
We have time for questions. I'll get us started maybe. I know you look at a lot of different nanoparticles, the effect of the charge, positive or negative, the size. What do you think is the most important property in the nanoparticles taken up by this?
Well, these polystyrene nanoparticles, I do believe that they are dependent on what nanoparticles we're talking about. For these polystyrene nanoparticles, we saw this huge difference between the effects of the charge, and also Franouche, she has a poster. When she looked at the effects of these particles on MDCK cells, which is the kidney cell lines, she saw a huge difference between the effects of the charge. The positively charged go 200 times faster than negatively charged particles. It doesn't mean that if we look at the silica particles, we're going to get the same. That's the way that some part of this research is going. For polystyrene nanoparticles, for alveolar epithelial cell monolayer, I would say charge matters a lot. But I cannot generalize it for all the nanoparticles and all other cell lines and different cell models.
Any other questions for Nazanin? Well, if not, let's thank Nazanin again. Thank you. Our next speaker will be Portia Peters from the laboratory of